Okay. So today we're going through chapters 25 to 28. So it's quite a lot. Actually, hold on. I'm gonna open my chat so I can actually see what's going on. Okay. Okay. So first is chapter 25, which is the history of life on Earth, how life originated and how it developed. So, first, the conditions on early Earth made the origin of life possible. So the first cell was hypothesized to be formed after 4 steps. So first, small organic molecules such as amino acids or nucleotides, they were synthesized with energy from the environment. So obviously, like atoms and molecules and elements, all of those existed. They just had to be synthesized into organic molecules. Then these molecules would aggregate to form macromolecules, which get packaged. So these macromolecules are just bigger organic molecules, which is easy to form. And then those get packaged into protocells, which will eventually develop self-replicating molecules within them and also its own machinery. So there are a lot of hypotheses for the synthesis of early organic compounds and where they came from. So it's possible that there was hot water vapor that resulted from meteor bombardment in early Earth, which condensed and allowed organic compounds and nutrients to form from the extreme heat. Also, UV energy and radiation from the sun and also outer space could have helped to synthesize complex molecules from the simpler ones. So two scientists who are Miller and Urey, they experimented on this. So they created environments that were similar to early Earth and found that this environment was capable of creating amino acids and other organic molecules just by themselves with similar energy levels and everything. So another hypothesis is that the first organic compounds were produced in hydrothermal vents, which are very hot underwater vents, or alkaline vents, which is very basic underwater environments. And between these two vents, the alkaline vents are more likely because the pH was more ideal for life than having extreme heat. So after the formation of the first organic molecules, macromolecules could have formed spontaneously without catalysts or other enzymes. So this could have been from dripping RNA or amino acid solution onto rocks and allowing for natural synthesis. So next, the formation of protocells is done by basically allowing vesicles to naturally form from lipids. So a protocell is here on the bottom left. It's basically a ring of a lipid bilayer. So when you put these lipids together, because they have a hydrophilic and a hydrophobic end, they will and if you put that in an aqueous environment, they will basically arrange themselves into a bilayer. And also there is a thing called Montmorillonite clay, which is here. This type of clay can also help with the natural formation of vesicles from lipids. Okay. And these are self-replicating, can grow in size and absorb RNA and other molecules. So part of the reason why it can self-replicate is because there can be substances that can go inside the protocell, and because of osmosis and water pressure, that cell will just keep expanding and will eventually have to divide because the membrane might fall apart. Yeah, so it can also absorb RNA in this way. Okay. So next is self-replicating RNA, which is called a ribozyme. So the first genetic material was very likely RNA because it did contain genetic information like DNA, but it can also function as a catalyst, which is the ribozyme, as we have noted here. So this was because the base pairs on RNA can hydrogen bond with themselves and basically fold the RNA into a specific 3-dimensional shape and can self-replicate because it can also act on other RNA molecules. So, very similar to natural selection, the ones that replicated the fastest were more present and favored. So any mutations that allowed for faster replication of RNA will be retained. And yeah. So RNA was the first self-replicating genetic material. And protocells could also absorb RNA from the environment, like I've said before. And this allows for further expansion of the cells because a higher intracellular concentration of RNA would allow for osmosis of water inside the cells, which causes the cells to expand. So as time went on, RNA could have provided a template for DNA synthesis because DNA was obviously more preferred because it was more stable. And yeah. Okay, so next is the fossil record. So fossils are preserved rock remnants of dead animals and plants and organic beings. They mostly accumulate in sedimentary rock since since that type of rock is made of very small grains that can be easily packed into different shapes. But some animals can be preserved in amber, which is tree sap, such as bugs or mosquitoes, and also ice, like mammoths. And there were also humans that were preserved in ice. So it's important to know that the fossil record can't tell the complete story of all of Earth's organisms and their evolution because a lot of animals don't die in the correct places to be fossilized, and others have also been destroyed or may not have been discovered and categorized yet because there are many, many layers of the Earth, and we've only found so many fossils. So fossil records can also provide a lot of useful information, which includes when the organism existed. So this is done through fossil dating or radiometric dating. So radiometric dating is based on radioactive decay and elements half-lives. So organisms, when they are alive, they will intake a variety of carbons. So they will intake carbon-12, which is a stable form of carbon, and also carbon-14, which is the radioactive carbon. And these are incorporated through nutrients and used in organic matter. So when these organisms die, the carbon-12 is stable, so the carbon-12 concentrations will not change. However, carbon-14 is radioactive. It's unstable, so it will radioactively decay to nitrogen-14. And there's no more carbon intake, so these concentrations will change without any extra input of carbon. So carbon-14 has a very specific half-life. So it's possible to find out how long ago the organism died by comparing ratios of carbon-14 to nitrogen-14. Okay. Fossils can also provide information about how organisms evolved physically. Since there are some similarities and differences between early reptile and early mammal bone structures. So for example, on the bottom here, all three of these bone structures have a hinge and a bottom jaw and also a temporal fenestra. So although these skulls did change physically, many of the parts are preserved. So that shows a physical evolution of these organisms. So this is just a chart of everything of the whole history of the Earth. So approximately 4.6 billion years ago, which is 4,600 million years ago, Earth formed, and then I'll be talking about all of these stages, but basically, so the first prokaryotes existed, the eukaryotes appeared, and the animals developed, the dinosaurs, and then the dinosaurs died off, and then more mammals appeared, and then here we are at human age. Okay, so first was the development of single-celled organisms. So the first single-celled organisms were prokaryotes because they are relatively simple, like they're not as complex as eukaryotes. So we can prove the existence of the first single-celled organisms as prokaryotes because they formed stromatolites, which are thin layers of rock that form when prokaryotes bind the thin layers of sediment together with their with a thin membrane film. So here is a image of a stromatolite that formed. And they also commonly form in these like piles on top of water. So yeah. Okay, next is photosynthesis and the oxygen revolution. So some of these prokaryotes that first emerged were photosynthetic and were able to use light energy to split water, which created their own energy and released oxygen as a byproduct. So these prokaryotes were largely water-based, and so the oxygen that they released would dissolve in the water. So at one point, since a lot of them were using photosynthesis, the oxygen got to a really, really high concentration, so it eventually leaked into the atmosphere from the water. And the presence of oxygen and a huge increase in oxygen could also be verified with the presence of iron oxide in old rocks. So iron oxide is basically when oxygen reacts with iron to form rust. So rust has its signature red color. So the rust can be in older rocks and which have red streaks in them. So about 2.5 billion years ago, there was an oxygen revolution where oxygen levels suddenly shot up in about 500 million years. So since these prokaryotes that already existed were initially living without oxygen, so they were anaerobic, these high levels of oxygen, which were initially a waste product, they were toxic and inhibited a lot of their enzymes and damaged their cells. So as a result, prokaryotes either died off, they had to live in anaerobic environments elsewhere, or they had to adapt and eventually use oxygen in cellular respiration. So this time period is when cellular respiration first develops. Okay, next is the emergence of the first eukaryotes. So eukaryotes are generally more complex than the prokaryotes because they contain nucleuses instead of nucleoids. They have membrane-bound organelles. They have ER and mitochondria. And they also have the ability to engulf a lot of other cells or materials by changing their shape. So this ability to change shape was actually an advantage because it allowed for endosymbiosis, where large cells were able to engulf smaller cells that evolved to become organelles like mitochondria and chloroplasts. So the engulfed cell, which is basically called the endosymbiont, which coexists in a mutualistic relationship with the larger cells. So the endosymbiont will live in this cell and is protected from the environment. And in exchange, the ones that we commonly talk about, which include the mitochondria and the chloroplast, they will provide nutrients and energy such as ATP and sugars for the larger cell. So another part of endosymbiosis was that the mitochondria had been engulfed and evolved in symbiosis before plastids such as the chloroplast. And this could be shown because all eukaryotes contain mitochondria, but only photosynthesizing cells will contain plastids such as the chloroplast. Okay, so endosymbiosis was also widely supported with cellular evidence. So first, the mitochondria and chloroplasts have separate division processes. So in a cell, they actually divide by themselves and they don't have to undergo cellular they don't just divide during cellular division. Also, endosymbionts have their own machinery and genomes. So as you might have already learned, the mitochondria and chloroplasts have their own separate DNA. And also they have separate membranes and transport systems. So the endosymbionts will have their own membrane and also a layer of the larger cell's membrane when it was engulfed in a vesicle. Okay, so here's a diagram of endosymbiosis. So initially there was an ancestral prokaryote that underwent a lot of folding to basically create the endomembrane system, and that those cells eventually engulfed smaller prokaryotes such as aerobic bacterium that formed the mitochondria and also other photosynthetic bacterium that formed the chloroplasts in the cell. So this was an ancestral photosynthetic eukaryote, and if it wasn't photosynthetic, then it wouldn't engulf these photosynthetic bacterium. Okay, so next was the origin of multicellularity. So the first multicellular organisms are small red algae, which I will be talking about later today. So this was a very, very big change in organism biology because it basically shifted all of the organisms from being single-celled to being multicellular organisms, which provided a lot of advantages, such as avoiding predation in groups, helping with specialization. So some certain cells can produce can just undergo photosynthesis, for example, while the other ones can undergo cellular respiration to help each other aggregate energy. And they can also integrate more environmental cues and resources. So we will be talking about a type of protist that will gather together in groups when it wants more food. So these groups can help them help the cells get more resources. Okay. Next is the Cambrian explosion, which basically involved the sudden appearance of a lot of animal phyla. So this included sea sponges, which are here, cnidarians, which include sea stars and their relatives, and also mollusks. So this Cambrian explosion also its most significant portion was aspect was the emergence of larger predatory organisms with adaptations to hunt and self-defend, for example, like long spines and body armor. So if you look at sea stars and the mollusk here, they have hard shells on the outside, and the sea star also has spines on top. So as compared to before the explosion, the organisms were only soft-bodied grazers. So this evolution of adaptations to hunt and self-defend helped with their survival and also eventual domination of organisms. Okay, next is the colonization of land. So the movement out of water was associated with adaptations that allowed for reproduction out of land without dehydration. So the first organisms to move out were algae that basically evolved, I guess, into plants because so for example, the waxy cuticle layer on plants prevented excess evaporation of water, which is a very good adaptation for moving onto land. And also plants could have colonized land with fungi. So fungi would form mycorrhizae with plants, which are basically root associations to help obtain more nutrients and also provide nitrogen for the plants. And the most widespread colonizer eventually were the arthropods, which included insects, bugs, everything like that, and also tetrapods, which are four-legged organisms. Okay, next is plate tectonics. So this is just a little bit about plate movement. So plate tectonics have a really large role in ancient speciation and the distribution of species in different continents. So plate tectonic theory basically says that continents are big moving pieces of the Earth's crust on a hot underlying mantle. So this supports the idea of Earth being multi-layered with a thin, fragile crust, a very big mantle that takes up approximately 80% of the Earth's volume, and also an outer and inner core at the center, which are the hottest parts. So the mantle also contains a lot of magma, which becomes lava when it erupts in volcanoes. So since these plates can move randomly, they will come into collision with each other. So the borders of the plates that come into contact with one another will commonly have earthquakes, volcanoes, or mountains because of the frequent shifting of the land. For example, the Himalayas formed from the Indian and the Eurasian plate here, so they are about there. And we also live very close to the Pacific plate here. So that is why we have a lot of earthquakes. Okay. And since these plates can move, they were originally one big continent called Pangaea, which eventually broke apart. So that's also why certain continents look like they can fit together. For example, the South American East Coast looks like it could fit with the African West Coast because they were hypothesized to be together originally. So when Pangea divided, species that originally existed together were divided, which led to fossils of the same species being in different continents, new species forming as the group spread only with each other and restricted the gene pool, and also fundamental changes in species as they were exposed to new environment conditions that they had to adapt to. Okay, so mass extinctions are also a part of the history of the Earth. So extinctions can happen for many reasons, which include a drastic change in the environment or a destroyed environment. So in the modern world, actually, a lot of species go extinct because of destroyed environments, like because of human action and destruction of forests and stuff like that. So really dramatic changes to the environment can lead to mass extinctions, which is basically when a large number of species worldwide become extinct at the same time. So this is different from a species extinction where only one species die out. So there were 5 big mass extinction events, and the most important two are the Permian and the Cretaceous extinction events. So during the Permian extinction, about 96% of the marine life was altered. And this also included a very active volcanism period that led to a detrimental chain reaction in species populations. And next was the other one is the Cretaceous extinction, which eliminated all dinosaurs except for birds, and it was likely caused by an asteroid or a comet collision. And this was supported by the presence of iridium in a lot of the soil, which is very rare on Earth, but very commonly present on asteroids and comets. So this was the extinction that basically allowed a lot of mammals to prosper. So consequences of mass extinctions include altered evolution and ecological communities. So a lot of species will die out and be replaced by other species, and also adaptive radiation, which I will talk about in the next slide. Hold on. Okay. So adaptive radiation is basically periods of evolutionary change when organisms actually they don't form new species per se, but they do fill ecological niches of other species that are no longer present. So so adaptive radiation can be worldwide or regional. So in Galapagos finches, they have actually undergone filling of ecological niches because for example, some of the finches mostly feed on trees, some others will mostly feed on the ground. So another example is basically of a species of animal that lives primarily on the ground and uses resources from the ground. If they die out, species that live on the trees will take over the niche on the ground and try to survive off ground resources. And they do this to minimize competition between itself and other species in its original niche. So species do this to minimize competition and minimize already existing competition. Okay. So next is the development of effective developmental genes. So developmental genes, they had a very significant effect as species changed over time. So first, heterochrony is the change in timing of developmental processes. So for example, bats can have earlier and longer growth rate of fingers than humans, so they will end up with very long fingers that make them look like they have wings. While humans, they don't grow fingers for that long and that fast, so we don't have that. So also, while human and chimpanzee fetuses have very similar skull shapes, they have different developmental processes and timings that are associated with the development of each facial part, which makes the adult faces drastically different. So in this, it's possible, for example, that the chimpanzee develops the jaw for longer and earlier than the human, so that's why they end up with a longer jaw. Okay, also different species may reach reproductive maturity at different rates. So there could be an evolutionary need for this because perhaps certain species have shorter lifespans or are more hunted and have to reproduce earlier. So for example, certain insects can be sexually mature at a couple weeks, while humans, biologically, they take until puberty to become sexually mature. Okay, next is pedomorphosis, which can result from heterochrony, and this is when the adult form of an organism can still contain juvenile characteristic parts. So this results when the development of reproductive parts happen faster than the other organs. So an example of this is the axolotl, which is a type of salamander. So usually salamanders don't have gills, but axolotls will retain their gills due to pedomorphosis. And also, changes in homeotic genes will change basic morphology. So homeotic genes basically determine what part of your body develops into what when you are grown up. So this is the Hox gene gene 7, and it is a homeotic gene. So in ancient ancestors, they developed into a large body segment with legs, but in modern day insects, they don't have legs on that body part anymore, so that's how they evolved and changed morphology over time. Okay, any questions? We can go on to the next chapter. Okay, next is chapter 26, which is the phylogeny and the tree of life. So phylogenies basically show evolutionary relationships. So species are usually named with binomial nomenclature, which incorporates the genus and the species. So for example, the panther's scientific name is the Panthera pardus. Panthera is the genus and pardus is a species. So so these organisms are classified by hierarchy. So these hierarchies are shown here, with the domain at the very top being the most broad hierarchy, going down to kingdom, phylum, class, order, family, genus, and species. So for example, for the panther here, they belong to the animal kingdom, the chordate phylum, which means that they have a spinal cord. Mammals, they're mammals because they have fur and they produce milk. They're carnivores, they're felines, and then eventually panther, and the very specific scientific name. So common ancestry can also be shown through phylogeny trees and not just by nomenclature. So on phylogeny trees, each branching point would be where a common ancestor should be. So for example, for the American badger and the European otter, they have a common ancestor at this point. And all of these species here have a common ancestor at this point. So sister taxons will share an immediate common ancestor. So for example, the coyote and the gray wolf are sister taxons because they share an immediate common ancestor here. So also rooted trees show the common ancestor of all taxa on the very left. So this here is a rooted tree because the common ancestor of all of these taxa is shown on the very left. So basal taxon will, this is a lot of terminology, but basal taxon will diverge from all other taxa. So here the leopard, oh, it's not a panther, it's a leopard. Okay, the leopard is the basal taxon because it diverges from all of the other taxa, and it doesn't have common ancestors with any of these other than the furthest one here. So also, just in general, two sister taxa can be rotated at the common ancestor. So these don't have to be like badger on top, otter on the bottom. They can also be otter on the top, badger on the bottom. It doesn't really matter. Okay. Okay, I think you should do this. It's a good practice. So, if you guys are still here and paying attention, which of the trees here shows an evolutionary history that's different from the other two? Just pick A, B, or C and message me in the chat. Great, thanks for the responses. So, the answer here is C is different from the other two because, like I've said in the previous slide, hold on. The two sister taxa can be rotated at a common ancestor. So here, you can see A is on the top, B, C, D, right? But then for B, these three taxa are simply like, so this is basically, first of all, flipped around, and then these three are rotated, and then these three are also rotated on the inside. However, in this phylogeny tree, it is shown that A is still the basal taxon, but then D diverges earlier than C or B, which is not the case. Because in these, C and D have a more common ancestor. So these have the most common ancestor, but in this tree, C and B have the most common ancestor, which is different from what the other two trees show. Okay. Okay, so phylogenies can be inferred from morphological and molecular data. So morphological and molecular homologies are physical and genetic similarities because of shared ancestry. So this is different from analogy, which is from convergent evolution. So here's just a really quick reminder of the difference between homology and analogy. So for analogous structures, from analogy, they don't have very close common ancestry, but the same features evolved due to the same need. So these streamline appendages are evolved in very different species. One is a fish, one is a bird, and one is a mammal, but they all evolved for the same purpose of swimming faster. But homologous structures basically have the same common ancestry. So all of these organisms have a radius and an ulna on the forearm, which are shown here. Okay. So molecular homologies are similarities in genetic material or segments of DNA that are also resulting from common ancestry. Okay. Oh, this is dense. Okay. So shared characteristics are used to construct phylogenetic trees. So the practice of cladistics is basically determining where to place species based on common ancestry to create a phylogenetic tree. So species are placed into clades, which include the common ancestor and all of its descendants. So taxon, which we have talked about, taxon is like each like group of organism. So it's only equal to the clade if it's monophyletic. So monophyletic basically means that it only contains one taxon and no other descendants. So if I can find one here. So for example, here the lancelet would be monophyletic because it doesn't contain any other relatives, I guess. So paraphyletic, on the other hand, it contains the ancestor and some of its the monophyletic contains the common ancestor and all of its descendants. The paraphyletic group will contain the common ancestor, all of its and some of its descendants. So this only contains 3 of its descendants. And the polyphyletic group will contain the common ancestor and all of its descendants, but some of them are not directly related to others. Like for example, so the common ancestor is here. These A, B, and C are more closely related than D is, but they have all come from the same number 3 common ancestor. Okay. So shared ancestral characters are characteristics that originated in a common ancestor of a taxon, which then separates the taxon from the rest of the phylogenetic tree. So shared ancestral characters are shown here. So for example, the vertebral column developed here, which separates everything else, separates everything from the lamprey down from the lancelet. And the hinged jaws will separate the base and everything down from the lamprey. So those are shared ancestral characteristics. And then, yeah, okay. My bad. Okay, so in polyphyletic groups, the common ancestor is not included. And then shared derived characters are evolved specifically in a clade. So that's the difference. And some other terms, an in-group is the group that we're studying, and the outgroup is a species or a group that is from the same evolutionary lineage as the in-group, but are not as closely related to the group that we're not studying. And so there are two ways to infer phylogenies, which are maximum parsimony and maximum likelihood. So maximum parsimony will infer the most explainable events, which basically include the least evolutionary events, and also maximum likelihood will identify the tree that is most likely to have produced a given set of DNA in an organism. Okay. Okay. So while physical similarities between species can help identify what the phylogenetic tree looks like, molecular similarities in the DNA can also help define evolutionary history that can't be physically seen through physical characteristics. So DNA especially for ribosomal RNA and mitochondrial DNA will evolve rather slowly because these features are similar in all cells. So orthologous genes in orthologous genes, the homology will occur as a result of speciation with divergence of genes. So there's an ancestral gene, but then once speciation happens, these orthologous genes will be slightly different because these are obviously two different species. And this can only occur after speciation events. So another is paralogous genes, where the homology will result from gene duplication and divergence. So these paralogous genes are similar to the ancestral gene, but they will be slightly different because they have been duplicated and changed after a lot of generations. Okay, molecular clocks can help track evolutionary time. So basically, molecular clocks use the constant rates of evolution in certain genes to estimate the absolute time of evolutionary change. So this is determined by plotting the number of mutations versus the divergence time. And it is good to calibrate the graph with a gene that has a very stable rate of change. So it basically shows you how many mutations accumulate over time, and then you can use that to kind of infer the mutations that will result after more time. So these usually show evolution of neutral genes because harmful genes are usually eliminated within a couple million of years. So yeah, these neutral genes will be preserved. Okay. Okay, horizontal gene transfer can impact the genes that get passed on vertically. So for example, the gene for metabolism in yeast is more similar to the gene in bacteria than archaea, although by lineage, eukaryotes are more related to archaea. So horizontal gene transfer can occur through transposable elements and plasmids, viral infection, and fusion of organisms. So anything that basically moves one organism's genetic information into another. And horizontal gene transfer is very important and prominent because about 80% of the prokaryotic genes have moved between species at some point during evolution. Okay, next is bacteria and archaea. Okay. Okay. Do you have any questions? So, okay, so first let's talk about the structural and functional adaptation of bacteria. So bacteria will have a lot of cell surface structures that help it evolutionarily and to infect organisms. So they have so on the cell surface they have peptidoglycans, which are modified sugars cross-linked by short polypeptides, which include which encloses the entire bacterial cell and has anchor elements that help stick out from the bacteria. So Gram stains are used to determine the composition of a bacterial cell wall. So a gram-positive bacteria will have a simple peptidoglycan cell wall, as shown here, and a gram-negative bacteria will have a complex cell wall that is composed of lipopolysaccharides, which are carbohydrates and lipids bound together. So gram stains are very effective when diagnosing patients with bacterial infections because gram-negative bacteria have lipopolysaccharides that usually are more toxic to the body, and they can also develop resistance faster than gram-positive bacteria since the outer membrane can prevent entry of a lot of drugs. Okay. Okay, and on the outside of bacteria, they usually have an outer sticky layer called the capsule, which helps it stick to substances and other cells and can also protect against the host immune system. So to withstand harsh conditions, bacteria can produce endospores with their chromosomes, which helps them reproduce. So endospores are basically a copy of the bacterial chromosome enclosed in a multi-layer structure. So all the water will be removed from the structure, which creates a very resistant spore that contains bacterial chromosomes. So the cell will then break apart and release this endospore, which is super, super durable and can survive for centuries before rehydrating and allowing more bacteria to form. So next structure is, oh, my bad. Next structure is fimbriae, which are short hair-like structures that help bacteria stick to surfaces. So they are shorter and also less in number than pili, which helps two cells stick together for gene transfer during conjugation. Okay. Also, about half of prokaryotes are capable of taxis, which is movement away and towards a stimulus. So most do this with flagella, which are long whip-like structures that propel the cell forward. So these flagella can be on one end or scattered around the cell, and each cell can have multiple of them. So eukaryotes can also have flagella, but the composition of eukaryotic flagella is very, very different from prokaryotic flagella with completely unrelated proteins. So, for example, sperm can have flagella that move them forward. And also, their bacterial cells have a unique genetic organizations, which includes a nucleoid space where all of its genetic information is stored, and also plasmids, which are basically smaller rings of genetic information that get transferred during conjugation between two bacterial cells. Okay, so bacteria can reproduce very, very fast. So that leads to even faster rates of mutation. So according to Campbell's, the mutation rates can be up to 8 million mutations per human host per day. So that obviously leads to very fast adaptation to environments. So this can lead to very fast resistance of bacteria to antibodies, since a small number of these bacteria will have mutations that make it resistant to the antibodies, and that requires more antibodies and different types of that antibacterial medication to treat the bacteria. So bacterial cells can also have even more genetic recombination from transformation, transduction, and conjugation. So genetic recombination is basically where genomes from more than one organism combine to create more diversity. So first type of genetic recombination is transformation, where the genetic makeup of the cell is altered by uptake of external genetic material from the environment. So these bacterial cells can basically pick up DNA fragments, RNA fragments from the environment and incorporate them into their own genome. The other type is transduction, where bacteriophages will carry genetic material from one bacteria to another. So during transduction, basically, a bacteriophage will infect one cell, inject its genetic material into the cell, and when it reforms, when the new bacteriophages reform, they might have parts of the old bacterial genetic material inside of them, and so that will be transferred into other bacteria. And then the third way is conjugation, where genetic material is transferred between two bacterial cells. So during this process, there will be sex pili that form between two bacterial cells, and basically the genetic material will go through that pili into the other cell. So, and this genetic material is not the actual nucleus chromosome, it will actually be the plasmid. And I think I have a slide on this later. So what determines the formation of these pili? So fertility factor, which is called the F factor, will determine the ability to form sex pili to interact with other cells. If you don't have this factor, you can't form the sex pili. If you have the factor, you can. And also another factor that's important is the resistance factor, or the R factor, and this allows for resistance to certain types of drugs. So the R factor allows for resistance to be passed on in different cells. Oh, okay. This is transduction, what I was just talking about. So, oh, that got cut off. So basically there will be a bacteriophage which infects the bacterial cell, injects its genetic material into the cell, and when it reforms, it's going to have part of the genetic material of the original bacteria in it, and then that can be incorporated into the new host cell. And then this is conjugation. So basically, one part of the plasmid will be transferred over to the other bacteria, and it will be replicated, and then they will have the same plasmid transferred eventually. Oh, so here's also the F positive and the F negative cells. So the F negative cells will not be able to form the sex pili, but F positive cells will be able to. Okay. So prokaryotes can have a lot of nutritional and metabolic adaptations. So first is oxygen metabolism. So obligate anaerobes have to use oxygen for cellular respiration, but obligate anaerobes cannot use oxygen for cellular respiration, or else they will die. So so this is what I was talking about 2 chapters ago. So initially prokaryotes could not use oxygen, but they had to evolve to be able to use oxygen. So obligate anaerobes can live by fermentation or by anaerobic respiration, which involves extracting O2 from substances that contain O2, which can be substances like nitrate ions, which are NO3-, and sulfate, which are SO4 2-. So during nitrogen metabolism, certain species can convert atmospheric nitrogen, which is in the form N2, into dinitrogen into usable forms, and this is called nitrogen fixation. So after nitrogen fixation, the usable nitrogen can be incorporated into amino acids, and also this nitrogen fixation has a very large impact on other organisms such as plants that need the nitrogen to grow. So bacteria, for metabolism, bacterial cells can group together to cooperate in metabolism. So a specific example here is the Anabaena. I don't know how to pronounce it, but here are pictures of them. So they have genes for both photosynthesis and nitrogen fixation. So so some bacteria, which are the heterocysts here, they can only fix nitrogen, while the others, which are all of them here, can only photosynthesize. So both types of bacteria can benefit from this relationship. And they can also create biofilms that can recruit other cells to join and grow bigger colonies. So these colonies are not multicellular organisms, they're still individual cells, but they just live together to cooperate. Okay, so there are a lot of diverse lineages of prokaryotes. So in bacteria, there are proteobacteria, which are gram negative. So they include the alpha, beta, gamma, delta, epsilon bacteria. They include the chlamydia, which are gram negative, which are parasites that have to have a host. They include spirochetes, and cyanobacteria, which are gram negative, which cyanobacteria use photosynthesis and might have components for nitrogen fixation, like the bacteria example we just talked about before. And there are also gram-positive bacteria, which can cause a lot of diseases, and also includes mycoplasma, which are extremely tiny bacteria without cell walls. So mycoplasma are extremely hard to get rid of. You can't just boil them and they die. They actually take a super, super long time to die, and extremely high pressures. So yeah. And archaea include extremophiles, which live in very, very extreme environments. So these include halophiles that live in very salty environments. And the protein and cell walls of halophiles allow for high survival in high salinity. So these halophiles will actually die if salinity drops too low. And they also include thermophiles, which live in extremely hot environments. So these thermophiles have adaptations that make their genetic material and protein stable at high temperatures. So Taq polymerase, which was used in PCR, is taken from thermophiles since the polymerase is heat stable and can be used in the extension step. And also methanogens are also a type of archaea which produce methane waste when oxidizing H2 with carbon dioxide. Okay. I don't think we can get through chapter 28, but I'll try to finish 27. We might. Okay. So certain chemo heterotrophic prokaryotes can function as decomposers, which basically break down different chemicals and substances. So they could break down dead organisms and other waste products to unlock important sources of carbon, nitrogen, and other environments that would otherwise be trapped in an unusable form. So prokaryotes and also they can also immobilize certain nutrients in the soil. So there is a complex relationship with soil and plant nutrients and also prokaryotes, but they can also convert forms of molecules into others for usage by other organisms. So for example, nitrogen fixation from atmospheric nitrogen, and also production of sugars and oxygen. So prokaryotes and its hosts can also engage in ecological relationships, which is called symbiosis. So there are three main types of symbiosis, which are mutualism, commensalism, and parasitism that can occur between prokaryotes and hosts. So in mutualism, both organisms will benefit from the relationship. So for example, this image shows a mutualistic relationship between the fish and the bacteria, because the bacteria reside in the fish's eyes, which produces a natural glow in deep sea environments. So the fish can use that to attract prey, eat it, and then the bacteria can gain more nutrients from the fish. So the next type is commensalism, where one organism benefits and the other isn't very affected. So for example, the bacteria living on your skin get a place to live in your pores and on your skin cells, and you provide them with food, but you're not very negatively or positively affected. So the last type is parasitism, where the parasite prokaryote will basically eat away part of the host organisms or take its nutrients, which will then harm the host. So the parasites that cause diseases are called pathogens, and it's important to know that parasites don't immediately kill the host, unlike a predator, but will take a while and drain its nutrients slowly. Okay, so prokaryotes can also have significant effects on humans. So mutualistic bacteria can live in our gut and help digest components that we can't. So they can synthesize certain carbohydrates, vitamins, and they can also help break down a lot of food components. And certain types can also stimulate human cells to secrete antibacterial components that target competition. So that's how we are benefiting the bacteria that live in our gut. So pathogenic bacteria can also cause disease by poisons. So exotoxins are proteins excreted by pathogens which cause disease, such as cholera. And that's caused by an exotoxin which stimulates intestinal cells to release ions into the intestine, which causes osmosis out of the water. Osmosis of water out of the cells. It causes diarrhea. The other type is endotoxins, which are lipopolysaccharide components of gram-negative bacterial cell walls. This is what I mentioned when I said gram-negative bacteria can cause disease. So they're only released when the bacteria die and the cell walls will break down. And horizontal transfer of genetic material can also make harmless bacteria more harmful. So E. coli was originally harmless, but strains that caused bloody diarrhea were horizontally transferred to them. And so that made the E. coli harmful. There are also bacteria defense systems such as CRISPR-Cas9, which involves using a guide RNA to recognize DNA, cut the DNA, and let the DNA self-repair to change gene expression. So that has been used a lot in biotechnology and to change expression of cells. And also, it can be used in prokaryotes can be used in bioremediation. Yeah, I know. I'll finish this chapter. And bioremediation, where prokaryotes remove harmful substances from the environment and produces nutrients instead. Okay. No, I need to go right now too. I have a lot of work today. I can't. Yeah. It's okay. The next chapter isn't that bad. It's just a lot of names. Okay. So, we'll finish here today then. Yeah, I have work.